Hi guys, and welcome to our first online lecture. So hopefully this works. We're going to be working on it, so if it looks a little weird, the sound is a little weird, the lighting's a little weird, I'm going to be fine-tuning all of this in the next upcoming weeks, hopefully upcoming days, and so we should all be back to normal and working pretty well. So let's get started on mitosis and meiosis. So this is what we are on right now. We're on chapter 8, cellular replication. Okay, so we've talked about cells, we know what's going on with the different types of cells, we know the organelles, we know that we're made up of cells, but now we're actually going to be getting into how this happens via cellular replication. So either mitosis or meiosis. Hopefully you guys remember these things. If not, we're going to remind you guys right now. Clicker. All right. So first off, when we're talking about cellular replication, we're talking about cellular division. And why do cells need to divide? Okay, so we have our little cell right here. Why would it need to replicate itself if it's already one cell? Right? Well, remember that when we were first conceived, right, when fertilization first happened, we had the sperm, we had the eggs, they got together. When you have that fertilized egg, you are a single cell. That single cell is then going to replicate via cellular replication, turn into 2 cells, then 4 cells, then 8 cells, and 16 cells, and 32 cells. This is how we become the multicellular organism that we are today via this cellular replication. So it's very important that these cells replicate so that we can grow. Another thing is, say if I were to cut my arm, right, my arm becomes damaged. Those cells there become damaged and die. So I don't have a gaping hole in my arm the entire time, right? My cells need to regenerate so they can repair any damaged, lost, or broken cells. Okay, so if I were to injure myself, you need to replicate cells to create new cells so that your body can, maintain itself. Also, when we're talking specifically about meiosis, this is where we get into the reproduction part. So we know we are made of part of our mom and part of our dad, right? It's not your dad's cells replicating and your mom's cells replicating and then they all of a sudden combine, right? It's your dad's sperm and it's your mom's egg that get together. So when we do this, this is known as meiosis. This is a form of sexual reproduction. So this is also another type of cellular division, which has to happen before we can actually get fertilization and get us. So when we start off talking about cellular replication, we're going to talk about the simple cells, those prokaryotic cells. Right? Remember that pro means before, karyot means nucleus or kernel. So these guys do not have a nucleus, right? Before the actual nucleus. So their form of cellular replication is very simple. You start off with what's known as the parent cell, and you end up with 2 daughter cells. And again, sorry sons, these are just daughter cells. There are no sexes involved here, right? But you have a genetically identical copy of the parent cell. So these 2 daughter cells are genetically identical to this parent cell right here. So what you see right here, we have our cell wall, we have our plasma membrane, you have that DNA in the nucleus. In the non-nucleus, it's known as the nucleolus or the nuclear area because these guys do not have a true nucleus. And what they're going to do is they're going to replicate their DNA because, again, we need 2 copies, one for this cell and one for this cell. So we replicate 2 copies of our DNA, okay, and then we're going to grow and we're going to split. And as we split, we become 2 separate individuals. These are 2 what are called daughter cells. And that's pretty much it for binary fission. Binary means 2, fission means splits apart. So this is very simple fusion where you take 1 and you end up with 2. The key here is that they are genetically identical to the parent cell. There's no difference. Now, when we talk about eukaryotic cells, this is where we're going to get a little bit more complicated here. So when we talk about eukaryotic cells, there's 2 different types of replication. The first one is known as mitosis. And mitosis is where you're going to replicate what's known as the somatic cells. Okay, so somatic cells are known as body cells. This would be my skin cell, right? I damaged my arm, I've cut myself, now I need to replicate just a body cell. My arm is not going through sexual reproduction right now, right? I'm not making sperm or eggs out of my arm. I just need to replace those damaged or lost cells here in that cut on my arm. So this is going to be for the somatic cells or the body cells. And in this case, very similar to binary fission, we start off with 1 parent cell, they replicate their DNA, and we have two daughter cells genetically identical to the parent cell. Okay? So that's what we see right here. Two daughter cells, which are both diploid, which we're going to get into in just a second. The haploid cells come later when we're talking about meiosis. So we have, actually, let's talk about this now. So this is known as diploid. Diploid means two, or di means two. So diploid means you have two copies of each of your chromosomes. Now we are made up of humans of 46 total chromosomes, but it's total because we have 23 pairs. Okay, 23 times 2 will give you that 46 total chromosomes. Okay, so we are known as diploid. All of my body cells, all of my somatic cells are all considered diploid. When we're talking about meiosis over here, this is where it's going to get a little bit different. We're talking about your sex cells. So they're no longer somatic cells, not body cells. They are sex cells. And our sex cells are our sperm and our eggs, respectively, whether you're male or female. Okay, so in this case, we want to go from that diploid, meaning an entire complete copy of all of our DNA, to something that's haploid. Haploid means half of the number. Instead of 2 copies of each chromosome, we have 1 copy of each chromosome, which is important. If you had an exact copy of one, say the egg, you had a full copy of that DNA from the egg, from your mom, you would essentially just be a little clone of your mom, which would be super weird, right? So we don't want that. You want half of the number, half of your DNA from your mom and half of your DNA from your dad, so that when they combine, it is genetically unique and you. So that's what we see here in meiosis. We see one parent cell replicates the DNA, but then splits twice. So we have 2 different splits, meiosis 1 and meiosis 2. Okay, when we're done with meiosis 1 and 2, we end up with 4 daughter cells, not 2. So mitosis, we had 2 daughter cells that are genetically identical to the parent. But for meiosis, we have 4 daughter cells that are genetically unique. Unique from one another and unique from the parent. They have half of the number of chromosomes that they should have, because the other half is going to come from, say if this is your mom, the other half is going to come from your dad, or vice versa. Say this is your dad. Okay, so you always want half the number of chromosomes, so that when you combine half and half, you get a whole, and that whole individual is you. That's why you're not genetically identical to either one of your parents. Essentially, you're kind of a blend of both of your parents. And we're going to talk about that in just a second. Now, we have to talk a little bit about some terms to know what's going on here. You're going to hear a lot of terms that are really, really similar. Chromosomes, chromatid, chromatin granules, sister chromosomes, homologous chromosomes, centromeres, centrioles. You're going to hear all these words and they all sound almost identical. So make sure that you can distinguish between the different ones, okay? So let's start with a couple of terms here. So first off is we have our DNA, which is going to be packed down into what's called a chromosome. Okay, chromosomes are essentially little packets of DNA. And that's because remember that your DNA, this double helix up here, is really long. It's really long, it's really complicated, and it can break, right? You don't want part of your DNA gone, because then maybe all of a sudden you don't have an arm, right? You don't have the DNA for an arm. It's not exactly how that works, but you can kind of think of it like that. You're missing DNA. You can't miss DNA. We need that DNA. We need all the DNA. So what your body does before it goes through before it goes through cellular replication, what it does is it tightly packs itself into this little chromosome. And what happens is you have your DNA double helix here, and it starts coiling up. It starts wrapping around itself, wrapping around itself, wrapping around itself. And then it's going to take these little proteins known as histones, and it's going to wrap around them. And as it wraps around them, it makes these little packets. These little packets, or these little balls right here, are known as nucleosomes, okay, little packets. And then you're going to take those nucleosomes and you're going to wrap those up, and you're going to wrap those up, and you're going to keep coiling, keep coiling, keep coiling, keep condensing, and further and further and further until you keep spiraling, and then folding and folding and folding, until finally you have this very condensed, very packaged, tight little package known as our chromosome. Okay, so first up is our chromosome. Okay? Now, this chromosome has all of your DNA in it. right? We have 23 pairs of these chromosomes. Okay, but we need a complete copy because we're going to be starting with mitosis first. And really, guys, if you understand mitosis, meiosis is going to come so much easier, but we really, 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 really got to understand mitosis first. So, in mitosis, remember I'm working with those somatic cells, I'm going to make identical copies of two cells. So I have my starting cell, my parent cell. I need to replicate that DNA because I need one whole copy of my chromosome to go to one cell, and I need one whole copy of my chromosome to go to another cell. So I need two copies. So what we're going to do is when you replicate them, essentially you're just making an entire copy of this guy right here. So we make that copy. These are known as replicated chromatids. Okay, so we have chromosomes, chromatids. I'm not going to be very particular on the verbiage. If you want to call it a replicated chromosome, you can call it a replicated chromosome. That's fine. These are also, because scientists like to name everything 2 or 3 times, these are also known as sister chromatids. Okay, they're sisters because they're identical, which isn't exactly a great nomenclature for it, but essentially what they did is one copied into 2. Now it's one complete copy of my chromosome here and another complete copy of my chromosome here. Okay? So, where they connect right here in the middle, this guy is known as a centromere. Okay, that centromere is kind of like this non-coding region of your genes, your DNA, that it doesn't matter if something's pulling left way, one way or the other, it's not going to get damaged. And that's because eventually, remember we have one complete copy of my chromosome here, one complete copy of my chromosome here, eventually they are going to get split apart. They get split apart right there at that centromere. Okay, so this is going to get pulled apart to pull one complete copy this way and one complete copy that way. Now, before we actually wind up all of our DNA, it's kind of found loose, right? Remember, it's this DNA double helix that's just kind of loose and floating around your nucleus. It's protected inside the nucleus, right? But it's still just kind of loose. So before it coils into a chromosome, it is known as a chromatin, not chromatid, chromatin granule. So you'll see chromatin, DNA chromatin, chromatid, or chromatin granules. All of them are the same thing. They just mean the exact same thing. Okay. Now, remember, this is essentially what we have our 23 of. Okay, this is a chromosome. Say it's chromosome number 1, right? We have chromosome 1 all the way to chromosome 23. Say this is chromosome number 1. Okay, we have one copy that we got from mom, and we have one copy that we got from dad. Put them together, we have that full copy that is us, right? Which is why you have part of your genes from mom and part of your genes from dad, because you actually got one of their chromosomes. You got part of their DNA. Now, if I was to go through and replicate, so say mom's DNA now needs to make another cell, so her copy needs to make a copy, and then dad's copy also needs to make a copy of himself. So he makes a copy too, because again, we're getting ready to split into 2 more cells. So we have our replicated, again, sister chromosomes, or sister chromatids, because again, this copy is identical to this copy, right? This is from mom, this is from dad. So these 2 right here would be known as sister chromatids. Here's where it gets a little bit confusing. If we have one copy from mom and one copy from dad, these are known as homologous chromosomes. Homologous meaning they're the same, these are the same, right? Same chromosome, this is still chromosome number 1. One side from mom, one side from dad, still where we got our 2 copies from. But they are considered homologous chromosomes because we got one from mom and one from dad. Okay, so sister chromosomes, right here, sister chromatids, they're the exact same. Homologous chromosomes, one copy from mom, one copy from dad of the same gene. We're still talking about gene number 1. We're just talking about our mom's side and our dad's side together, which would make it the homologous part. So far, so good? Remember, if you guys have any questions while we're going through this, I will have a live stream discussion during our lab section. So fingers crossed, and that all works pretty good too. So we're gonna work through this, guys. We're gonna work through this. Now, here are your 23 copies of each chromosome, right? You have copies 1 through 23. In fact, your 23rd pair is known as your sex chromosomes, either your X and your X if you're a female, or your X and your Y if you're a male. Okay, so this is a single chromosome right here, right? We have 2 copies, one from mom, one from dad. Sister chromosomes because they are exactly the same. Homologous chromosomes because one came from mom and one came from dad. But these 2 are sisters because they're identical, the exact same. Okay, so this is how we can actually get kind of a blending of these different traits, because we get some traits from mom and we get some traits from dad. Sometimes these are dominant and one will kind of outshine the other one, and it will show up despite that the other one is still present. We're going to talk about all that when we get to patterns of inheritance in the next lecture. Now, when we talk about cell replication, really what we're talking about is when we're talking about cell life, we're talking about 2 phases. Okay, you're either going through replication or you're not. If you're going through replication, you're known as the M phase or the mitotic phase, meaning you are replicating. If you are not replicating, you are in what's called interphase, or the phase in between replications. 90% to 95% of the cell's life is spent in interphase. It's not replicating every second. You don't want your cells to just continually replicate. Actually, if you do, that's how you get things like tumors and cancers. It's uncontrolled cellular replication. Your cells just keep replicating. That's a bad thing. So our bodies keep our cellular replication under wraps and in check. So most of the time we're spent during interphase, but interphase is still a very important phase. 1, it's living its happy little cell life. And 2, it's getting ready for replication. If we need to copy our DNA, it needs to be done in interphase before we actually get to the replication part. If we need to copy some of the organelles that we're trying to replicate, remember, like the mitochondria, the Golgi body, the rough and smooth ER, all those things you're going to need for your new cell. So in interphase, that's when all these things get made. So interphase is very, very important, but it's actually not part of mitosis or meiosis, and we're going to talk about the steps of that later. So interphase, most of the cell's life. Mitotic phase, that's where we're actually going through the division. Now, there's a couple spots, there are a couple different parts of interphase. First up, we have G1 phase. So this is M phase, this is mitotic phase. So imagine we just finished replicating. So we enter here, we're a brand new cell. We have all the DNA that we need. We are nice and diploid, remember, 2 copies of each chromosome. So we are complete, right? We're gonna live our happy little cell life. So this first phase is known as the G1 phase. And this stands for growth 1 phase. Imagine if we had one big cell and then it split off into 2. Those 2 smaller cells are smaller than the original parent cell. So the first thing the cell needs to do is grow nice and big. He needs to grow to the same size as the parent cell. Because if he didn't grow, every time we did a cellular replication, right, we'd split smaller and smaller and smaller and smaller and smaller and smaller and smaller. That wouldn't be good. So we need to grow back up to that normal, healthy, big cell size. That's what G1 phase is. So growth 1, growing bigger as a cell. The second phase is known as S phase. This stands for synthesis phase. And remember that synthesis means to create. So if we are creating, in the synthesis phase, we are creating DNA. This is a very important step. This is where we're actually replicating our DNA. Remember, we need 2 copies, one for this cell and one for this cell. The only way we can do that is by copying our DNA. And we're going to talk about how we copy our DNA later on in the semester. But right now, you just need to know that it does get copied. One for this cell and one for this cell. So that is what S phase is doing. It's doing this duplication, right? Replicating our chromosomes and our DNA. Next up is the G2 phase, and in G2 phase, it is the second growth phase, but this time, we're growing those organelles that we need. We needed more mitochondria. Let's grow some mitochondria. We need more Golgi body. Let's make more Golgi body. All the things that your cell is going to need for when it's off on its own, that's kind of what G2 phase is. We're making all those important organelles that the cell is going to need when it moves out on its own. Then finally, we enter the M phase here, the mitotic phase, meaning either mitosis or meiosis, cellular replication here. And that's where you take 1 and you split into 2. Important here that we've already replicated our DNA. We've already copied it. Because once we enter here, we need 2 copies. Because all M phase is, essentially, is the splitting of the 2 cells. Okay, so we need everything already replicated before we get to that part right here. Finally, the last phase is known as cytokinesis. Cyto meaning the cell or the cytosol, the fluid on the inside, right? And kinesis means the split, we're just the splitting of. So technically, this is the physical separation of the 2 cells. And we're going to talk about the difference of that and M phase in just a second. So, remember, in interphase, it's spending most of its life in this phase, right? Most of its life just living its happy little cell life, growing the organelles that it needs, replicating the DNA that it needs for the actual cell replication. Okay, so it's doubling everything that it needs, it's growing in size, and it's living its normal and happy cell life in interphase. Once we get to the M phase, or the mitotic phase, it too is divided into several steps. Okay, we have the mitosis stage, which is where we actually get the cellular division, right? They're splitting, they're breaking up the chromosomes, they're moving apart. And then finally, the cytokinesis, just where that cytoplasm, right, fully separates. That's kind of the very last step. Mitosis is basically complete, and all it is in cytokinesis is just popping those 2 cells together. So it's really, really quick stage. Now, here's where we're going to get a little complicated, and obviously I can't have you guys do this what I would normally do in class, which is draw this for me, but I would practice it by drawing it out. So take a piece of paper out, draw out every phase, right? Then you'll really understand what's going on. And trust me, if you really understand mitosis, meiosis will be so much easier later on. All right, so reminder where we are. We are in interphase, right? We're not in mitosis yet. Not in mitosis yet. Interphase is the normal, happy little cell life. So in interphase, right, we have these chromatin granules. Remember, the loose, just kind of flowy DNA. It's not packaged up tight into those little chromosomes yet, right? It's still that loose, flowy DNA. Okay, it is protected in the nucleus right here. Okay, remember that everything is in that nucleus to be protected. You don't want it getting out, you don't want it slipping away, you don't want part of your DNA being lost or broken. You need it all in that nucleus. So it is protected here in what's called the nuclear envelope. Now, in the first stage of mitosis, so we're finally starting mitosis. So we are no longer in interphase, we're no longer living our little cell life, right? We are not in the form of chromatin granules here in this nucleus anymore. Now, we're getting into that chromosome. So, the stages of mitosis are the following. Okay, and you will need to know these. Easy way of remembering this is PMAT. Prophase, metaphase, anaphase, and telophase. Okay, these are the only 4 stages in mitosis. Okay, interphase is not one of them, nor is cytokinesis. Just prophase, metaphase, anaphase, and telophase. So these are the ones that you guys will need to know, and these are the ones you will be responsible for your test. So, starting with the very, very first one, and I will give you guys a whole step-by-step breakdown of what's actually going on here. So, in prophase, pro means, remember, before, right? This is the first phase, so before or the first phase. Okay, so in prophase, you're getting ready to do division. Now, we had inside of our nucleus right here, we had all this protected DNA, right? This loose DNA. We don't want that anymore. Now we're getting ready to split. So now what we have to do is we have to coil up and we have to condense into those tiny, tiny little chromosomes. Remember those little X's, those little chromosomes. This is because we're going to be getting ready to split. So, in interphase, step 1, this nuclear envelope starts to break down. Okay, it starts to go away. Step 2, these chromosomes are actually going to condense. They're going to coil up into those little X's. Okay, this is really important because these are our little packets of DNA that we don't want to lose. Okay, so step 3 is we have what's called these little centrosomes. If you hear me call them centrioles, centrioles and centrosomes are essentially the same thing, so you might hear me use either one. Okay, so we have these centrosomes right here. Centrioles, yeah. Centrosomes, centrioles, same kind of thing right here. What they do is we have 2 copies of them. One is going to one cell, the other is going to the other cell. So, in this case, what we have right here is they start to migrate towards the outsides of the cells. That's because this side is going to pull away from this side. So step 3 is that these centrosomes, or the centrioles, migrate to the opposite ends of the cell. Step 4 is you have what's called these mitotic spindles. These mitotic spindles are kind of like, they're spindle fibers. They're also called spindle fibers or mitotic spindles. They're essentially what's going to pull the cells apart. So it's kind of made out of the same material as the cytoskeleton was, the thing that holds up the cell, right? But now we're used to pull the cell apart. Okay, so that's what these guys are going to do right here. Okay, so these spindle fibers are going to separate and start pulling the two cells apart. So this is early prophase. This is late prophase. So by the end of prophase, again, we've lost the nuclear membrane. It's gone. We have condensed chromosomes now, no longer chromatin granules. We have chromosomes, sister chromosomes, in fact. Okay, so step 1, we lost our nuclear membrane. Step 2, we condense our chromosomes. Step 3, these centrosomes move to the outside of the opposite ends of the pole. And step 4, we start forming all of these mitotic spindles, right? Connecting the two to start ripping apart these cells. Okay, so that was step 1. That was prophase. Prophase is one of the most important phases because it's getting everything ready to go. If you mess up how you get it ready, you're going to mess up the rest of the process later. Now, PMAT, right? Prophase, metaphase, anaphase, telophase. Prophase was first. We're done with prophase, right? Second up is metaphase. Metaphase is way easier stage. The chromosomes, the sister chromosomes, line up in a line, right? Sometimes called the equator, sometimes called the metaphase plate, right? They just line up in the middle. So we have our centrosomes on the outside because they're going to be pulling apart. And we have our chromosomes lined up in the inside. Now these chromosomes are attached at that centromere, right? So these mitotic spindles attach at the centromere because one side's going to get pulled that way and one side's going to get pulled that way, and then your little X is going to get pulled apart. One whole chromosome goes this way, one whole chromosome goes that way to each of the individual cells. So that is metaphase or middle phase. They line up in the middle. Next up is anaphase. Anaphase is another very simple phase. We have those chromosomes lined up in the middle, and now they're going to separate apart. So that's all. This is the physical separation of the two chromosomes. Metaphase, they line up. Anaphase, boom, they're separated one and one, going to either side of the poles. Either side of the cells, also known as the poles of the cells. Kind of like the north and south poles, the opposite ends. Same thing, opposite ends of the cell. Telophase is our last phase. So telophase is our last phase right here where you can actually see the opposite of prophase. So think about everything that happened in prophase. We lost our nuclear envelope. We condensed our chromosomes. So now what we're going to do, step 1 in telophase, we're going to bring back that nuclear envelope, right? Because step 2 in telophase is we're going to uncoil those chromosomes. We're now going to go back to those chromatin granules. We have that loose, flowing bit of DNA. So we're going the opposite of prophase. Prophase, we lost our nuclear envelope, we condensed our chromosomes. Here in telophase, we are going to uncondense our chromosomes, so we're going to bring that nuclear membrane back. Okay? And then finally, we have what's called cytokinesis, which is the physical separation of the two. Now, there are a couple important things to notice. One, when we're talking about animal cells, when they're pinching in telophase, right, they're going to be one cell, and now they're going to start to kind of gather and start to pinch and create a little kind of crease. This crease right here is known as a cleavage furrow, right? Furrow kind of means to gather, and cleavage, well, you know what cleavage means. Okay, so we're making one cell into two, so we're gathering it right there, right? We're gathering that plasma membrane right here, and eventually during cytokinesis, we physically separate them. So when you see them connected right here and right here, they are still in telophase. It is only once they have physically separated that they are now in cytokinesis. So telophase, cytokinesis. Right? You can see kind of right here in the middle, this is that little gathering of that plasma membrane because we're looking to separate the two cells in telophase. So this would be in telophase right here. Now this is only true in animal cells. In plant cells, because they don't have a squishy plasma membrane that controls their shape, they have a plasma membrane, but it's on the inside of a very rigid cell wall. So because it's on the very inside of the rigid cell wall, what happens is they actually lay down a little bit of new cell wall, splitting their two nucleuses, right? And they create what's called a cell plate. So animal cells have cleavage furrows, plant cells have cell plates. This is still telophase, okay, because the cytoplasms are still connected. It's not until you actually get that full wall, that full cell wall made, are you now in cytokinesis. So telophase, cytokinesis. Still connected, right? Just creating that cell plate because it's a plant cell, not an animal cell. Totally separate cytoplasms, hence cytokinesis. All right, we were gonna watch a video. I'm gonna let you guys watch this at home. Just type in crash course mitosis. It really will help. It shows you really a lot of animations, better than I can, and actually walks you through the steps. So there's a million different resources online that you guys can watch this. Pick whatever video you want, but I do want you guys to actually watch a video and see this stuff actually starting to split. Now, let's talk about meiosis. Remember, we just learned about mitosis in great detail. These are our somatic cells. We're making cells that are identical copies to us. Well, now we're getting into meiosis, and that's not what we want to do here. So imagine I have fertilization, right? I have mom's sperm and dad's eggs, other way around. Mom's eggs and dad's sperm, we get them together, we have fertilization, right? We are a diploid zygote. A zygote means just a developing embryo. Okay, so it's diploid because it has 2 copies of each chromosome for a total of 46. Because remember, we have 23 chromosomes. 2 times 23 equals 46. So that's where we got our chromosomes from. Now, as this cell grows, it's going to grow and grow and grow and grow. It's going through mitosis. It's just replicating its body cells, its somatic cells as it grows. But then, say we have a full copy of a new mom and a full copy of a new dad, and they want to have a baby. So what they're going to do is their sex cells, their sperm and their eggs, are going to go through meiosis, and they're going to produce haploid gametes. Gametes are sex cells. Gametes are sperm or eggs. So they're haploid, meaning only 1 copy of each chromosome instead of 2. Diploid, haploid. 1 copy of each chromosome. That's because half of it's going to come from the egg, half of it's going to come to the sperm. When we put it together, we fertilize it, we are 2N, we are diploid. Right? We have a complete copy that we need. So this is where we're going to go from a full diploid cell to now a haploid cell. Either a sperm or an egg, depending on whether you're talking about mom or you're talking about dad. So that is meiosis, and that's what we're going to talk about now. So in meiosis, we have essentially the same stages of mitosis, but they happen twice. So we go through the first division where we take the parent cell, we replicate the DNA, right, sister chromosomes, but now we have 1 full copy of mom and 1 full copy of dad. These are homologous chromosomes. So now we're talking about homologous chromosomes. When we're talking about homologous chromosomes, what can happen is because we have 2 levels of splitting, right, we have twice as much splitting, you can actually get 4 daughter cells that are genetically unique from one another and the parent. It doesn't show too much right here, but this is just kind of an overview of what we're going on. We're going to see that in just a second. So, remember how I told you, if you really, really, really understand mitosis, meiosis will be so much easier. Here's why. Because essentially, meiosis is just mitosis twice. All the stages are the same. Okay, remember interphase, that's not the phase where we're actually replicating, that's just living happy little cell life. But then we have meiosis 1. Meiosis 1 has prophase, metaphase, anaphase, and telophase. It's exactly the same. Okay? It's just called prophase 1, metaphase 1, or anaphase 1 because it's happening in meiosis 1. So, same thing happens, right? In interphase, this is regular cell life. We have our nuclear membrane right here. We have our chromatin granules on the inside, right? We have our centrosomes right here, just kind of hanging out. Then when we actually get to prophase 1, right, we're going to have everything the same happen. The nuclear membrane disappears, the chromosomes condense, the spindle fibers form, the centrosomes move to the opposite ends of the cells, exactly like normal. But here's a key difference. These homologous chromosome pairs, remember one from mom and one from dad, as they are getting ready, they're actually going to come so close to each other that they kind of overlap a little bit. As they overlap a little bit, these two sections of DNA get swapped. So now there's a little bit of your mom's DNA on your dad's chromosome, and there's a little bit of your mom's DNA, sorry, other way around, on your dad's chromosome. Okay, so now you kind of have a blending of mom and dad in each of the chromosomes. This is how we get what's called genetic variation. Part of your mom's traits, part of your dad's traits, but they get blended and you have this new assortment, this new mix. This is what drives things like evolution. Change over time happens because we get some traits from mom and some traits from dad, and we're able to blend them, right? Sometimes more crossing over happens than others. Have you ever seen kids that look almost identical to one of their parents? Oh my God, Stacy looks just like her mom. That's probably because she got, in this crossing over, she probably got the bulk of her mom's genes. Sometimes the kids don't look either like mom or dad. They definitely look like a blending of two. So that would be more even crossing over, right? You've got about half your mom's chromosomes and half your dad's chromosomes. This is very important, and this happens in prophase 1 of meiosis 1. So, crossing over of homologous chromosome pairs happens in prophase 1. This is very important for what's called genetic variation, right? Variation in the population when it comes to our genes. So, these little sites are crossing over right here. It's where mom's genes line up closely with dad's genes, and they do a little bit of swapping, right? They're swapping a little bit of DNA. He takes some of hers, she takes some of his. So now, in metaphase 1, it's exactly the same, right? They're going to line up on the equator that metaphase played. It's the center, essentially. But here's the only difference, and another important difference. In this case, we have pairs lining up next to each other. Before, we had individual sister chromosomes lining up on top of each other, which meant the X got split. One half went this way, and one half went this way. In this case, we have two X's which are next to each other, which means the whole X is going to go this way, and the whole X is going to go this way. So now what we're doing, instead of splitting up sister chromosomes, we are splitting up homologous chromosome pairs. You are essentially splitting up an entire double copy of your mom and an entire double copy of your dad. Okay, now remember, mom has a little bit of dad's DNA over here, and dad has a little bit of mom's DNA over here, but they are completely separate. So the homologous chromosome pairs split. That's what we see right here. This X is going to go up, this X is going to go down. It's not like the actual X's are splitting up. This pair of X's is going to go that way, and this pair is going to go that way. Hopefully that makes sense to you. That's the biggest difference between meiosis 1 and mitosis. Okay, we have homologous chromosome pairs which line up and do crossing over. And in metaphase 1 of meiosis 1, homologous chromosome pairs line up on the equator. So in anaphase 1, homologous chromosome pairs get split. Not the X's themselves, right? They don't go from 2 to 1 and 1. It goes from 2 and 2. So those homologous chromosomes get split. The entire X goes that way, the entire X goes that way. Remember, if you guys have questions on this, let me know. There's also a million resources, videos, etc. on this online that you can always go look up. Go watch Crash Course as well. There's another video for this one. Okay, so we have now exited meiosis 1. We have split out our homologous chromosome pairs. We still have sister chromosomes, right? Remember this little X on this side and this little X on this side. But now, right, you enter telophase 1, which is exactly the same, right? The cleavage furrow starts if you're an animal cell, cell plate if you're a plant cell, nuclear membrane comes back, chromosomes start to uncoil. Then we move right into prophase 2. Prophase 2, same thing happens. Nuclear membrane disappears, chromosomes condense, right? Spindle fibers start to form, centrosomes move to the poles or the outsides of the cells. And essentially now when you're in meiosis 2, you're going through mitosis, right? Sister chromatids line up on the equator, sister chromatids are pulled apart, you end up with a single copy of each gene in this one, right? Those X's have split, one goes this way, one goes this way. And now we end up with 4 daughter cells which are genetically unique, which isn't showing it right here. right? We have these two cells which are going to split, and now we have 1, 2, 3, 4. Because we had crossing over, each one of these little individual cells is going to have a different combination of, say, mom's genes and a different combinations of dad's genes. And this is how we get genetic variation, right? How even siblings can be genetically similar but look different from each other because they have a different combination based on this crossing over that occurred in meiosis 1, specifically prophase 1. I know this is a little bit confusing. Rewatch the video, it should help, and watch the crash course video as well. So that's exactly what you guys are here. Go see the crash course video. He's really great at this, and he's got a lot of great information about how they're splitting, why they're splitting, and the results of that. So, just a reminder, right? We have mitosis. This happens in body cells or somatic cells, like my arm cells, skin cells, all that, those different kind of cells, right? You have your duplicated chromosomes. Remember, we need a copy to go with one cell and a copy to go to the other. So each one of these little X's has been duplicated, right? In metaphase, they line up singularly in sister chromatids, and the X's get split. One side goes here, one side goes here. In meiosis, right, in prophase 1, we have crossing over between homologous chromosome pairs. Homologous, one from mom, one from dad. Little crossing over to get a little blending of the two, right? In metaphase 1, we have homologous chromosome pairs which pair up, not sister chromatids, homologous chromosome pairs, and one entire X goes on one cell, one entire X goes to the other cell. Then you enter meiosis 2, and those X's actually separate, and now you get one copy of each in each cell. And you can see right here, they are haploid. They are haploid after meiosis 1. Okay, diploid. By the time we're done with meiosis 1, we are haploid. In meiosis 2, we are still haploid. Okay, and this is what you can see right here, right? Moms with a little bit of dads right here, a different section of dads right here, a little bit of mom right here, a different section of mom right here. So each one of these individuals is genetically unique from one another and from the parent cell. Okay, so because this can happen many different ways, right? Say we had 2 blues on the right side and 2 reds on the other way. I'm looking at this backwards. 2 blues on the left side and 2 reds on the right side. Imagine if they line up like this. Maybe you would get most of dad's genes going over here and most of mom's genes going over here. So in this case, this is where, say this kid looks just like dad, right? He got most of dad's genes, so he's gonna look just like dad, like a copy of dad. Whereas this one maybe looks just like mom, right? Got most of mom's genes. Or, right, say you have this possibility, a red lines up with a blue, just totally random. Again, this is just kind of happening, floating around inside the cell. So in this case, we actually have a more equal blending of the traits. So maybe these kids look kind of like mom and kind of like dad, but not like these who would look mostly like mom and mostly like dad. So that's how you can get, again, some kids which look very similar to their parents, some kids which are more of a blending of their parents. So remember that crossing over creates genetic variation. This is all happening in prophase 1 of meiosis 1, and this is how we get all of these different potential variations down here. 4 individual daughter cells, all of them genetically unique from each other because of crossing over. Crossing over is so important for genetic variation and for mixing up of genes and traits and stuff, and giving us the mechanisms for evolution later on, which is why organisms have evolved from being asexual to being sexual organisms, because that potential for crossing over is really, really good. It's a really great thing. We also have a random fertilization of eggs, right? You never know which one of these sperm is actually going to be able to penetrate this egg and therefore fertilize it. You also don't know which one of the eggs that the females produce is actually going to get fertilized. So this is where it's kind of like, well, can't you predict what your kids are going to look like? You really can't, because of things like random fertilization, right? It might be something that future societies can do and kind of tweak and mess with our genetic codes and stuff like that, although the ethics might not be super cool with that one. But you never know which one of these, and again, you don't know what genes he's got, you don't know what genes he's got, right? All these little tiny sperm are going to have different combinations because of that crossing over. and therefore you don't know what kind of traits your offspring could have actually had. It's probably going to be some kind of blending, but it could be mostly mom, it could be mostly dad, it could be a combination. You don't know. And that's because of random fertilization and random, the chances of who's going to cross over where, who's going to get pulled apart to which cell is actually going to get a chance to fertilize. You never know. It's very, very random in that respect. And again, this is just a replicate of what I've just told you guys. And that is it for the cellular replication lecture. Hopefully that helped you guys. I know it's not the ideal situation. This is the first of the videos that we're going to do. And I know that there's some technical issues, and I'm sure there will be all semester long, but we're going to do our best. So I will be able to answer any questions that you guys have via email, via our live streaming feeds, whenever we get those going. But good luck to you guys, and hopefully this helps. Okay. You'll see me soon. I won't see you soon, but you'll see me soon. So I wish you guys the best of luck. I hope everybody's happy and healthy. And yeah, good luck to you guys. I'm here for you, even if I can't be, virtual hug. So, all right, guys, take care, and I'll see you soon.